Okay, so I'm going to start off this explanation by first explaining what the first section talks about, which is the ultraviolet and visible absorption in organic molecules. So what are organic molecules? Organic molecules are molecules or compounds containing unsaturated groups. So what are unsaturated groups? It could either be that or that or that. So these are all groups. They're all groups found in molecules, and if they were found in these molecules, said molecules, or certain molecules, that molecule would be considered organic. In addition to these groups that were just the unsaturated groups that were just said, and they're unsaturated because they have a double bond, basically. We have benzene ring. So benzene ring with alternating double bonds. These are all considered to be organic molecules. And these organic molecules, they could absorb in the ultraviolet or UV or visible light part in the spectrum. So as I just said, that these unsaturated groups plus this benzene ring or plus benzene rings, they could absorb light, or they could absorb in the ultraviolet or visible light part in the spectrum. Specifically speaking, I'm just going to put them in the side just to refer to them. So these are the alternating double bonds and benzene. These groups plus the benzene ring are known to be called as chromophores. So what are chromophores? Chromophores, essentially, they are organic compounds. So they are organic compounds with unsaturated groups and can absorb in the UV plus visible light part of the spectrum. So calling them chromophores essentially is just specifying these groups into an idea of them being called as chromophores instead of organic compounds, because organic compounds is a broader way of calling them. When we say chromophores, it's more specifically saying that these specific compounds or groups, they could absorb in the UV and visible light part of the spectrum. Okay, so now how do they absorb? How do they absorb in the UV light or the visible light of the spectrum? What do we mean by saying that? It's basically due to the electrons in the bond being excited to an empty orbital of higher energy. So from what we studied in previous chapters regarding the electron excitement charts, is that whenever an electron goes up and falls back down, so it went up to here maybe and then went back down, it would emit energy, some type of energy. And when it does that, specifically in our example now about chromophores, these electrons are that are being excited, which are called excited electrons because they're jumping up and down. Essentially, they're jumping to an empty orbital without an electron in that higher energy level. Another characteristics of these chromophores is that they're colorless. So that's another important point, is that they're colorless. Why? It's because the energy that is involved in this process so they're colorless because the energy involved in this process of absorption is high. So when it's high, with the fact that they're absorbing the UV and visible light of the spectrum, they appear to be colorless. And also from our data booklet in the IB, we could see that the specific wavelength that they absorb will give out a colorless, quote-unquote, color. However, examples like ethene, which absorbs in the wavelength of 185 nanometers, and in ethene as well, there are alternating CC single bonds and CC double bonds. So CC single bonds and CC double bonds, these are alternating in ethene itself. And then what this results in is that this ethene molecule is going to have delocalization of pi electrons because of here and here, pi electrons. The delocalization of them being alternated. And when this happens, less energy is required compared to the absorption of UV light. And when this happens, the excitement of electrons is not that highly energetic. So the absorption in ethene occurs in the visible region. Another type of pigment are anthocyanins. So anthocyanins are pigments that are aromatic and they're water soluble. They're water soluble because of the amount of hydroxyl groups found in them, and because of the flavonoid C6C3CX skeleton. And they're mostly found in plants, hence why they actually help in photosynthesis, or the absorption of certain colors and the emission of certain colors. So when we draw the flavonoid C3C6 skeleton, it's a pretty complex skeleton, or a molecule found in anthocyanins. So we're going to have a ring here, and then oxygen right there. And another ring here. So we have 2 rings, and the C3C6C3 molecule is found here and also in these rings. So because of this, we could also explain how less conjugation happens in the structure of cyanidin, which is a type of an anthocyanin, specifically in an acidic solution. We could explain why it absorbs in the blue or green region and it transmits red light. So we could draw it as so. So again, these are water soluble, and we could realize that from noticing these OH groups and the rings. So as I said earlier, the conjugation in cyanidin, specifically an acidic solution, it's going to have it will transmit red light, and it will absorb the area between blue and green. Okay, so what happens if cyanidin is found in an alkaline solution? There's going to be more conjugation, and this will be shown in the blue color. So before we had a plus sign on the oxygen, now it's gone. Now we will remove this double bond here, and then what we will also do is remove the double bond here, creating a double bond right there, removing this hydrogen, and also removing this hydrogen but making this oxygen an O-, because we removed the positively charged hydrogen atom. And what we will also do is add a bond right there. And what this does is it will make it more conjugated, but now instead of absorbing the blue-green region, it will start to absorb the orange region, and it will transmit blue light compared to the absorption of blue light of when it was in an acidic solution. Now, the conjugation of pi electrons that were just drawn, they're the ones that give color, hence why the excitement of electrons occurs, as I explained in the section previously. And the more extensive conjugation of pi electrons, the result will be a longer wavelength. And when a longer wavelength occurs, the color absorbed or transmitted will differ. And this also affects the pH of the solution, hence why the alkaline and acidic solutions. And the last point that I want to point out is that the addition of other groups changes the polarity of the groups that are also already in anthocyanins. So if we add other groups that are not already in cyanidin, it will obviously change the properties. So moving on to the third type of biological pigment, they're called carotenoids. Carotenoids are lipid-soluble pigments, and they will harvest light in photosynthesis, so they also have a role in photosynthesis. They also contain a long hydrocarbon chain, plus the alternating single and double bond carbon-to-carbon bonds, as we could see here, 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 and also here. So these bonds will be alternating, and the specific shape of this vitamin A, specifically retinol, is derived from a Pauline chain containing 40 carbon atoms, which may also be terminated by cyclic end groups, which are here, or whatever these groups were. And they also may be complemented with oxygen-containing functional groups, such as the ones right here. Now, the hydrocarbon carotenoids, the ones that contain a long hydrocarbon chain, they're known to be called as xanthophylls. And examples of xanthophylls are alpha carotene or vitamin A, plus some other examples such as beta carotene. And because of these bonds right here, the unsaturated CC double bond, they can be susceptible to oxidation, so they could undergo oxidation. And because they undergo oxidation, they also can be catalyzed by light, metals, and hydroperoxides. And what this does is that it results in the change of color, loss of activity in vitamin A, or the bad smell of these molecules. So maybe when certain medications require vitamin A, and then a person maybe realizes that this activity or this efficiency of vitamin A is not that good, this process could have happened. Okay, now I will be explaining a concept called chromatography, especially paper and thin layer chromatography. So this is going to be basically the isolation object that is going to be used, maybe a bottle or a beaker, and this is going to be the sample of amino acid. However, the one in the bottom, is going to be the initial position of this amino acid, and then we're going to have two distances traveled by this amino acid, this distance right here and also this distance right here. Because it's in a specific solvent, the solvent is going to travel upwards, and we could also create an analogy of a flood taking everything with it in its path, the flood being the solvent here. So the solvent is going to go upwards, above or towards the top of the paper. And this section right here is called the solvent front. And because we have to remove the paper as soon as the sample gets to the solvent front, that is because the sample will almost fall off the other side if we did not remove it, because the flood is going to keep taking away stuff from its way, in its way. And as the solvent goes up, we're going to have, as I said, these two distances, and we could calculate the RF value by that. So the distance traveled by the sample, which is this whole distance right here, is going to be the numerator. And the distance traveled by the solvent, so the one right here, so how much it went up, is going to be the denominator. But what happens when we have two amino acids, different amino acids, with the same RF value? So what we do is that we will rotate the paper 90 degrees, resulting in this shape right here. And then, as we could see, amino acids are going to be in different positions, obviously, because we rotated 90 degrees. And then the solvent is going to be here, and the solvent is going to be going up. This is the solvent front right here. This is the distance traveled by the sample, and this could be the distance traveled by the solvent. And obviously, since different distances will happen or will be resulted in, different RF values will occur, and we could take the second RF value to be applied to that amino acid. And then we have something called the stationary phase. It's when the water contained in cellulose fibers in the paper, when that occurrence happens, it's called this stationary phase, where we could point out, or when we could realize that the cellulose fibers contain water, and then they're in the paper. And then what thin layer chromatography is that it uses a thin layer of solid on inert support to basically undergo this paper chromatography, basically investigation or whatever, the calculation of RF values for different amino acids. Separation also occurs separation occurs more by partition with water as the stationary phase. So separation will occur more if it was occurring in the stationary phase, with water, obviously. And the choice of solvent depends on the polarity or other particular pigments, as could be seen here. This specific example was that the chromatogram was used in a second solvent with a different polarity, because if we use the same polarity, that might result in a very similar RF value, which is inaccurate to our specific investigation. And each of the separated components can be recovered pure by a spray or a dye spray dye. Another point is that the section containing the component is scraped or scraped off the glass and then dissolved in the solvent. So the section on the paper containing the component is going to be removed from the glass and then dissolved in the solvent again. This will be basically a cause or an effect of avoiding inaccuracy. And then the solution will be filtered to remove the solid support, and the solvent can then be evaporated to leave just the pure component that we need. So these are all the points that I've just mentioned are the steps that it takes when removing the paper and how we purify the component to get very accurate results for our values. Now I'm just going to summarize basically the chlorophyll and heme or heme section, because it's kind of complex and it requires general knowledge of certain molecules or groups, and it would take time to draw them all out. Now, chlorophyll and heme, they are porphyrin compounds. Porphyrin compounds include examples such as hemoglobin, myoglobin, chlorophyll, and other cytochromes that are chelates of metals with large nitrogen-containing microcyclic ligands. Now, we studied this in the chapter 3 HL section, and ligands usually replace water in certain processes. Now, the cyclic system occurs in CP3 hybridization as well, and these porphyrin compounds absorb light energy and they could undergo redox reactions compared to the other biological pigments that I have just explained that may undergo oxidation, such as the electron transport chain, because it's moving electrons across. So it's moving electrons across protein channels until it gets to the. Basically, this is the way the ions are going to go. Firstly, then water ion is going to go here, oxygen is going to leave here, and the hydrogen over here will create water compounds. Additionally, hemoglobin, what it does is that it carries oxygen from blood to other parts of the body. So it's basically an oxygen carrier. It's a protein. And one of the quote-unquote blood disorders that occurs in some people is called thalassemia. So thalassemia is basically the disease that happens when someone has a very low or below average below a necessity value of hemoglobin molecules, which then results in weaker stamina because they're losing more oxygen molecules per unit time. And this happens because hemoglobin is responsible for carrying oxygen, and then if there's less hemoglobin, less oxygen is going to be carried to other parts of the body. Now, low pH and high pressure of carbon dioxide cause oxygen to be released, obviously. Because low pH and high pressure of CO2, they will result in O2 released. And when O2 is released, obviously this process of releasing it will have to do with hemoglobin, because hemoglobin is responsible for carrying oxygen. And finally, temperature. If there's high amounts of temperature or high values of temperature occurring in reactions or that certain part of the body, more oxygen will be released, because we know that temperature and pressure and volume, some of them are inversely proportional to each other and some of them are proportionally to each other. Proportionally to each other means that when one of them increases, the other increases as well. And inversely proportional is when, for example, volume increases, pressure will be decreased. And this is an example of inversely proportional processes.